Self-incompatibility requires glycosylphosphatidylinositol or GPI-APs remodeling by the poppy post-GPI attachment to protein 1 ortholog HLD1. We studied the self-incompatibility poppy system and how it triggers programmed cell death. Because poppy flowers have pollen and stigma mature at the same time, these plants developed a system to avoid self-pollination. It depends on two determinants, PGAP1, a novel transmembrane protein expressed in the pollen, and its cognate PRSS secreted in the stigma. The interaction of both cognate determinants triggers programmed cell death. The same occurs in transgenic Arabidopsis plants expressing PRPS and PRSS. It means Papaver's self-incompatible system makes Arabidopsis thaliana self-incompatible. But what's happening between the determinants interaction and the pollen tube death? Which genes are participating in this response? In this paper, we show the results from a forward genetic screen carried out using transgenic self-incompatible Arabidopsis thaliana plants. Plants restoring the normal self-seed set were selected. A candidate affecting the male gametophyte was spotted for restoring a normal self-seed set and called HLD1. Whole genome sequencing identified Arabidopsis thaliana PGAP1 as the causal gene for HLD1, and it was confirmed by CRISPR-Cas9. It's a functional ortholog of the mammalian post-GPI attachment to proteins PGAP1 gene, encoding a protein involved in the deacylation of GPI-anchored proteins. This deacylation is required for the self-incompatible programmed cell death, resulting in no seeds development when PRPS and PRSS are expressed in the pollen and stigma respectively. On the other hand, the absence of PGAP1 expression reveals a normal seeds development even if both determinants are expressed in the flower. In addition, this deacylation is required in self-incompatible like programmed cell death in ectopic expression. The phases involved in the GPI biosynthesis have been established in mammals and yeast. The nascent immature GPI-AP undergoes further modifications during the transport from the endoplasmatic reticulum to the Golgi and then to the plasma membrane. Using antisense oligonucleotide approach, we observe that PGAP1 does not just work in the transgenic self-incompatible Arabidopsis context, but that it's required for SI-induced pollen tube growth inhibition and cell death in Papaver. Find all the details in Current Biology, Lin et al., 2022.